Lab 5A and Lab 6 have the most components for setting up, so give yourself plenty of time to do so. For Lab 5A, it is essential that the competent cells remain frozen at -80 or -20 degrees Celsius without thawing as much as possible. When conducting the lab, keep the competent cells on ice. Teachers have acquired ice from the school's athletic trainer or nurse. Some sites provide manual ice crushers for teachers to use. Regardless, this lab requires the cells to be kept cold. Should students need to remove the cells to add reagents, such as plasmid or LB, remind them to hold the microfuge tube by the rim, away from the competent cell solution. Contrary to cold cells, there are a few reagents that actually increase transformation efficiency if at room temperature. We suggest bringing the liquid LB that is added to the P+ and P- tubes, as well as the plates themselves, to room temperature. Just as you did for Laboratory 2A, you want to set and calibrate your water bath to 42 degrees Celsius the day before the lab in preparation for the Lab 5A heat shock. Classroom flow is also important. To avoid crowding around the water bath, you may want to collect each group's tubes or have each group approach the water bath for the heat shock protocol one at a time. Lab 6 also has a lot of moving parts. The most sensitive piece of equipment is the column. Make sure that your columns are stored upright and are hydrated. A dried resin bed can be rehydrated, but it takes time for the resin beads to settle down. Additionally, it takes time for buffer solutions to move through the column. You may want to run down your columns the day before or the morning of the lab to ensure that only a few millimeters of buffer are above the resin bed. Finally, a quick reminder that both labs generate bacterial waste, so ensure that your students wear the appropriate PPE and have all been informed of proper waste disposal instructions prior to conducting the labs.